Hello everyone, my name is Rahul. In the previous video, we had discussed about obtaining the foreign gene product. In this video, we are going to discuss about downstream processing. Downstream processing includes two processes. It includes two processes, namely purification, and separation. First, we will have to separate the products, then we will have to purify the products. So, after the completion of biosynthetic stage, that is, after the formation of our required product, it has to be subjected through a series of processes before it can be sold out in the market or it can be called as a complete product. So why do we need to separate the product? Because when we are using the recombinant DNA technology, it is not always possible to obtain a single product. So most of the times, mostly multiple different types of products or different quality of product is obtained from the biosynthetic stage. Why does this happen? Because we see that host cells are multiplying inside the culture, so it may be possible that one host cell is not able to achieve its maximum functioning, so it will produce a product which is of lesser quality and the other cell will produce a product of higher quality. So when we get the product, it is usually a mixture of various different qualities of the product. So to standardize our product, we will need to separate out all the different qualities, and then there will be a criteria upon which we will decide which product to keep and which product to not keep or discard. So first we will separate out all the products that are formed or all the different qualities of products that are formed. Second, we have to perform purification because because the product may be contaminated or there may be non-desired products along with the desired protein. So we will purify the product to remove out any contamination so that it can be safely introduced into the market. Next, what we do is addition of preservatives. Preservatives are chemicals which increase the shelf life of the product. So during downstream processing, preservatives are also added. Then all formulations have to undergo clinical trials and safety trials. So whatever product that we have obtained and we are trying to introduce into the market, with or without the addition of preservatives, they have to undergo clinical trials and safety trials to determine their safety, along with quality testing. So this strict quality testing is required for each product to ensure that a bad quality of product does not enter the market, because it may produce very harmful effects on the person or on the animal or on the organism on which the product is used. So the downstream processing and quality control testing, they vary from product to product depending upon what the product is used for. For example, recombinant vaccines, they will have different types of trials and different types of quality testing. And insulin production, which is also a recombinant type of insulin, it will have different type of downstream processing. So essentially downstream processing includes separation and purification along with clinical trials, safety trials, and quality testing. With this, we finish off with our discussion on downstream processing. See you in next video.